Welcome to Plenary Session. This is the video and audio edition. You're going to get audio on the usual Plenary Session feed. You're going to get video on YouTube. If you're listening on the Plenary Session feed, I'm doing my ASCO roundup. I'm going to try to cover all of the papers, although I'm getting a little bit tired. I'm going to try to cover all of the papers. If you're a fellow doing a journal club, this is going to be invaluable preparation, but I encourage you to tune into YouTube because I've got visuals, I've got graphics. The audio just isn't the same for this session. So go to YouTube if you're listening to the audio feed. We'll be back to pure audio sometime soon. Today we've got a big paper. We've got DESTINY BREAST 4. Trastuzumab deruxtecan in previously treated HER2 low advanced breast cancer. This is the talk of the town. I heard it got a standing ovation. People were standing up and clapping, and, well, you'll see what I have to say about it. I think this is a hard one for me. I spent a lot of time thinking about this paper, reading this paper. I even had my colleague, Timothy Olivier, he read it as well. He gave me some thoughts. I've pulled together a lot of this, and at one point you'll see how we both think the same. Let's hit it. All right, let's do it. You came here for this, and I hope it doesn't disappoint. My goal is that I hope I say something you haven't yet thought of. That's my only goal. But you're going to find that we all have our limits, and for a man who likes to look at data, when you don't show me the data, that's my limit. So let's hit this paper. Trastuzumab deruxtecan, previously treated HER2 low advanced breast cancer. This is the DESTINY breast trial. This is what it looks like. Obviously, you know this. You probably could draw this in your sleep. That's the kind of preparation we give in medical school, this kind of stuff, this hard-hitting, important stuff. Now, what do you see here? You see an antibody that's directed against HER2. And you see a cleavable linker that can be broken, and then you see the payload. And that payload is DX8951, which obviously you think about all the time. And this is a very potent payload. It is a topoisomerase inhibitor. Next slide. Anti-tumor effect of DX8951. This goes back to 1997. It was a good year back then. Still have a fondness for the music of that era. It's a novel analog, and it actually says somewhere in this that this is several times more potent than SN38. So, topoisomerase inhibitor, really, really potent. We'll come back to this, but this has been a payload that's been around for a while. Now it's tethered loosely to a trastuzumab-directed antibody. We'll come back to this. They busted out the new Ventana. This is the Ventana HER2. They said for investigational use only in their manuscript. This is what they're doing. They've got this particular immunohistochemistry assay. Now listen, I'm not an expert on the kinds of dyes you squirt on paraffin or fresh tissue. If you are, if you've got some thoughts, you're listening to Plenary Session, you're a fan of Plenary Session, you're a histopath person, and you know all the intricacies of the Ventana HER2 assay, you drop me an email. You can email me at my website, www.vinaykprasad.com, the contact me link. You tell me what you know, and if you got something that you want to say, you can come on the show and we'll talk about it. But this is the gist of it. We all know IHC3+, they're not eligible. They're considered HER2 positive. IHC2+, in this study, they look at that FISH ratio. If it's over 2, of course, that's also HER2 positive. This is the assay that they use. For a moment, I had a momentary thought. I'll come back to that. And basically what they're saying is if your HER2 FISH ratio is over 2, you're also ineligible. If you're IHC2+, and that ratio is less than 2, you're eligible. If you're IHC1+, you're eligible. IHC0, of course, you're not eligible. So this is the group in the middle. And by some estimates, it's a sizable chunk of people. It's like 40% of people. Now, the next in complexity comes in. Okay, this is a bit of a complexity. This study is expected to enroll, so many patients, physician's choice versus the new drug. After approximately 60 hormone receptor negative patients have been enrolled, further enrollment will be limited to subjects with hormone receptor positive disease. After approximately 240 hormone receptor positive subjects who have not had prior CD4/6 inhibitor have been enrolled, further enrollment will be limited only to subjects who have had prior therapy with a CDK4/6 inhibitor. So they want to make sure they've got enough people who have had CDK4/6 inhibition. They're going to do okay on that metric. They don't want to have too much triple-negative breast cancer. Okay, it's getting a little bit complicated, but this is it. HER2 IHC status will be stratified by this, that, the number of prior lines of chemotherapy, 1 or 2, and whether or not you received a drug like palbociclib. Okay. Investigators have to choose one of the control treatments for each subject before randomization. All right. They got to choose it up front before you get to know what you're going to get. The primary endpoint was, as God intended, progression-free survival. It's always progression-free survival. If only there was a moment in breast cancer history where we learned that progression-free survival wasn't a good endpoint in metastatic breast cancer. When was that moment? Avastin. I remember it well. Well, it is what it is. But this trial will also have an OS endpoint, and they looked at that as a secondary endpoint. Key secondary endpoints, PFS in all patients. Now, when you look at PFS in one group, and then you look at it not in the excluded group, but in everybody, this is called a nested subgroup analysis rather than an adjacent subgroup analysis. And the person you have to read on this is Sunny Kim and I. We've done 2 papers on this topic, nested versus adjacent subgroup analyses. Key secondary endpoints, PFS in everybody, overall survival in the hormone receptor positive cohort, then everybody. Secondary endpoints include investigator-assessed PFS, confirmed objective response, duration of response, etc., etc. So these are the primary endpoints, and they're going to end up hitting the ball out of the park for all of them, so we need not quibble. Okay, this is the rabbit hole that I fell down, because I like to really get a sense of who is in this study. What were the kind of prior treatments had they gotten? And then when they progressed on this new drug and they progressed on the control arm, what did they get? These are the questions that I always have when you have many, many drugs used in sequence and you're looking at the endpoint that I really care about, overall survival, which this actually does state. So I want to know what you got before, what you got after. I found 2 rules. Before I tell you the rules, I'm reminded many years ago, there's all these like little logic puzzles, and they say there's on one side of the creek there's Bob, Sue, Mary, Tom, Joe, and Bob and Sue can never be in the rowboat together, and Mary and Tom can never be left alone on a shore together, and the rowboat can only seat 2. How do you cross all the people across the river so that all of these rules are met and everyone gets from one side to the other? What are the steps? How many steps does it take? This kind of logic puzzle, and I'll tell you what, this starts to feel like that. The first rule: Eligible patients must have received chemotherapy for metastatic disease or had disease recurrence within 6 months of completing neo or adjuvant chemotherapy. Patients with hormone receptor positive disease must have received at least 1 line of endocrine therapy. That's right, from the methods. And then the protocol. Inclusion criteria number G. To be enrolled in the study, you has to be treated with at least 1 and at most 2 prior lines of chemotherapy in the recurrent or metastatic setting. If recurrence occurred within 6 months of neoadjuvant chemotherapy or neoadjuvant chemotherapy, the neoadjuvant chemotherapy would count as 1 line of chemotherapy. Targeted drugs such as mTOR inhibitors, PARP inhibitors, PDL1 inhibitors, histone deacetylase inhibitors, CDK4/6 inhibitors, and endocrine therapy on their own do not contribute to the count of prior lines of chemotherapy. They don't count as chemotherapy, although regimens with such agents used in combination with chemotherapy would still count as 1 line of chemotherapy. Whew, okay, now we're going to have some fun. This is where my head started to hurt a little bit. If you're hormone receptor positive, as the vast majority of people in the study are, you had to have gotten 1 line of endocrine therapy in the metastatic setting, because they're not counting the endocrine therapy in the adjuvant setting. You had to gotten endocrine therapy in the metastatic setting. And if you are hormone receptor positive or negative, you had to get at least 1 or at most 2 prior lines of chemotherapy. But if you recurred very soon after adjuvant chemotherapy, that counts. And then all those other drugs don't count at all towards lines of chemotherapy, but they could count towards lines of any treatment. We'll see. Okay. The rules, one more time. Hormone receptor positive, you had to get endocrine therapy. 1 to 2 prior lines of chemotherapy in the metastatic setting, could get cyclin 4/6 kinase inhibitors, and 80% did get it from the table, roughly. Hormone receptor negative, you had to get 1 to 2 lines of prior chemotherapy. Okay, the big table. Table 1. Now you can start to think about table 1, as I spent a great deal of time thinking through, and I'll walk you through it. There's a hormone receptor positive cohort, 331 patients getting trastuzumab deruxtecan, 163 patients getting physician's choice. All patients, 373 getting trastuzumab deruxtecan, 184 getting physician's choice. Now you're cooking with gas. Prior therapies, CDK4/6 inhibitors, 70%, 70%, and 64% in everybody. 64% in everybody. Well, of course, that includes the triple negative people. Immunotherapy, 3%, 4%, 5%, 6%. Endocrine therapy, 99.7%, 99.8%. 98.2%, 93%, 89%, chemotherapy, 100%, 100%, 100%. And the lines, median is 3, the range is 1 to 9. Okay, the number of lines of therapy, I didn't think about that 9. I got to think about that 9 too. Okay, we'll talk about it. Number of lines of therapy, 1, and this is lines of therapy for metastatic disease. 1 is 7%, 2, 25%, and 3 or more, about 2/3 of it. Okay, whew. Now we got to take these rules and apply it. Let's walk through this puzzle. Next slide. The first thing I'm going to do is I'm going to subtract out all patients minus hormone receptor positive patients to create what they should have presented, a triple negative breast cancer cohort, because these two are the two groups in there, and this should add up to the total numbers. And I was about to do this when I remember Timothy was telling me that he had been working on some table. And I said, let me look at Timothy's table. And it was exactly what I had been thinking about doing. And so I just took Timothy's table. This is Timothy's table. But, he's right. This is the first thing a good reader of literature would do. Subtract this out so we get a sense. Okay, now let's focus on the hormone receptor positive population. Hormone receptor positive. Okay, so they've gotten 4/6 inhibitors, and it's roughly, 70% of people. They've gotten some immunotherapy. I wonder why. They've gotten endocrine therapy, and they've gotten chemotherapy. And here's what it says. In the lines for metastatic disease, about 7% have only gotten 1 line. So how do you get 1 line of 1 line of treatment for metastatic disease, and you had to have gotten a hormone therapy, and you could only get 1 or at most 2 prior lines of chemotherapy, but you don't have 2. The hormone and the chemo is not 2, it's only 1. How does that work? And in my mind, I think the only way to solve this boat across the river puzzle is that these people had to have recurred after adjuvant chemotherapy within 6 months, and then their only treatment they got was endocrine therapy plus minus cyclin 4/6 kinase inhibitor therapy. That's all they got. They got dose-dense ACT, they recurred on month 4, and then they got, abemaciclib plus, endocrine therapy, or palbo plus endocrine therapy, or just endocrine therapy. And then after they progressed again, they immediately went on this study. That's the only way I can see it. And if you disagree with me, please put in the comments how you can get this 1 prior line of metastatic disease. Remember, adjuvant therapy, and they had to have gotten a chemo, and they had to get endocrine therapy. And the endocrine therapy has to count here as metastatic disease. The chemo could be the adjuvant therapy. I think this is the only way to solve the puzzle. Tell me if I'm wrong. So this is what these people got. Okay. The people who got 2 prior lines of therapy, I think they recurred. They got an endocrine therapy plus minus a cyclin 4/6 kinase inhibitor, and then they got chemotherapy. That's the most likely. So they got 1 line of chemotherapy, which is acceptable. The 3 or more, they only could have gotten at most 2 lines of chemotherapy. Okay, they only could have gotten at most 2 lines of chemotherapy, and then they could have gotten multiple lines of endocrine therapy, and they could have gotten cyclin 4/6 kinase inhibitor therapy. And so, they could have gotten endocrine therapy, progressed, and had that added, or they could have gotten, endocrine therapy plus palbo, progressed, got fulvestrant, for instance, and then progressed, and then got 2 chemotherapies, and then entered the study. Okay. But, now I saw that 9. I really got to sit down and think, how could you have 9, but you can only have at most 2 chemotherapies? You must have gone through the Rolodex of endocrine therapies and other drugs to get to 9 while only having 2 chemo. We'll have to think about that. I think this is fair. These are who these people are. Tell me if you disagree. Tell me if I'm missing something in my little boat across the river problem. Okay. Now the triple negative breast cancer cohort. Here's where it gets a little bit fun. Remember, I've subtracted these, so we got 42, 21. These are roughly the numbers. And what you see right off the bat, my first question is, 6 and 4 have gotten palbociclib or ribociclib or abemaciclib. Why did they do that? Why did they give them that? Does anyone know? Could anyone comment below what is going on? Am I missing something here? What's going on there? Immunotherapy. Now, of course, we do have Pembro, frontline, combination chemotherapy, CPS over 20. I think it's a bit low. I think it's actually supposed to be about 40% of triple negative breast. Tell me if I'm wrong here. Isn't that a bit low? But that's okay, Maybe they didn't always get all that pembro. I don't have a big problem with that. I just want to know, are we on the same page? We think that's a bit low? Endocrine therapy for hormone receptor negative breast cancer, 23%, 40%. WTF? Am I missing something? Is the math somehow wrong? Am I missing something here? What's going on? What's going on that endocrine therapy is being given here? What's going on? I'm really wondering. Am I missing something here? This is the only way the math works out. So and this has been triple checked. Okay, either something wrong with the math or something wrong with these docs, because I don't know what's going on here. Chemotherapy. They've gotten chemotherapy. What percent have gotten platinum? Okay, how many of these people have gotten PARP? But really platinum for this one. Have they gotten platinum? That's very active in TNBCC. And how many have germline BRCA? I'd love to know that. Okay, now here's another puzzle, okay? How do you get 3 or more lines in the metastatic disease setting when you could only be treated with 2 lines of chemotherapy and you're hormone receptor negative? How do you get there? All those other drugs don't count. Are we talking they got olaparib and then 2 chemo lines? Olaparib doesn't count, so that could get you to 3. Or are we counting that endocrine therapy, which, by the way, why did we give? So, I think there's a little bit of a puzzle here. Now, there's a way to solve the puzzle. What is that way to solve the puzzle? It would be called the appropriate reporting of what you're talking about here. Would it be so hard to tell me how many people had been treated in the adjuvant setting, how many people had de novo metastatic disease when you were treating the adjuvant setting, what were the treatments you got, how long between your adjuvant treatment and metastatic recurrence, how many people got what drugs, anthracyclines, taxanes, what do they get in the first line setting and the second line setting? Is it possible to give me some more granularity so I don't have to be, this logician puzzling over this? But I think, they're purposely, let's be honest, they're purposely reporting. They're not reporting the triple negative breast separate. They're reporting it all together or hormone receptor positive alone. That's intentional. So you cannot see these things. Because I think if you saw these things, if you saw this, you might wonder what is going on. You might say, WTF, what is going on with this pretreatment? You might say that. If you saw all these people hormone receptor negative getting endocrine therapy, you might wonder what is going on. And I think they don't want you to wonder what is going on. They want you to think, standing ovation, time for you. Okay, they want you to think that. So I don't think they want you to wonder. So I think this is an issue here. This is an issue because I really don't have a clear sense of what is going on. What did they get? What percent of these patients are de novo? What adjuvant therapies did they get? How many lines of endocrine therapy? How much cyclin 4/6 kinase inhibitor? How many prior chemos and which ones? Which ones did they get? Is that too much to ask? How can I deconstruct a paper when you don't let me have any of the facts? Okay, it's very difficult. Next thing I saw. Okay, in the physician's choice group, you get a 5-way choice, but you don't get an unfettered choice. Timothy has published a paper on physician's choice. They often get this restricted choice. When I go to a menu and I go and somebody invites me to dinner and they have a little card on the table so you can only order 2 things, I'm not happy. I want the full unrestricted menu. And similarly, I want that in my clinical trials, an unrestricted choice. And the company needs to just shell out the money and give people an unrestricted choice. But here they give it for eribulin, which is decent, but eribulin is typically a, 2-3 prior lines of chemotherapy in the metastatic setting. Then you give eribulin, Xeloda, nab-paclitaxel, gemcitabine, paclitaxel. I don't think that this is totally crazy, but I wonder, where's my carboplatin? I really do wonder, where's my carbo? Where's my anthracycline? Some of us like an anthracycline. It's a very potent medicine in breast cancer. We'll come back to that. Here is the main results, the PFS. 5 months to 10 months. PFS benefit hazard ratio 0.51. You see in the paper, they like to stretch it all the way out. But here I've put it in the proper aspect ratio. This is the right aspect ratio. And this is what the supplemental aspect ratio had for triple negative. So I have hormone receptor on the top, triple negative on the bottom. And even though the sample size is small, I don't think you see any suggestion that there's a differential interaction here. I think it's all consistent. There's a PFS benefit. I think so. Overall survival, and this is what blew them away, 17.5 to 23.9 months. And 8 months to 18 months in the triple negative cohort, in the hormone receptor negative cohort. I think, I keep calling it triple negative, but I'm just adhering to the old-fashioned way. I guess it's low HER2, hormone receptor negative. but it is conventionally a triple negative. It's not HER2 positive, but okay, whatever. You call it whatever you want to call it. Hormone receptor negative. I think there's a signal there, even though that it's a small population. I don't see any reason to suspect a differential treatment effect. But the moment I see overall survival, I want to know immediately what happened after people progressed. And you're taking me from about a 5-month PFS benefit to a 6-month OS benefit. It's getting a little bit bigger, and that makes me wonder a little bit about post-progression therapy. What is it? What is it exactly? I see that you are treating your hormone receptor negative patients with endocrine therapy. So what are you treating them when they progress? I mean, it seems to be a fair question to ask. The response rate of this drug, I gotta say, there ain't nobody can deny. It's got a good response rate. You got a good response rate, your new drug. 52%! Boom! I mean, it is an open label study. The only person blinded is the reader, me, because I don't know what they're giving before or after. But it's open label study. But that's good. I mean, that is good. And 16.3% investigator choice. I don't know. It's within what we say, 20% for, multiple lines in breast cancer, the hormone receptor positive cohort. I think that's reasonable. Here I've pulled some response rates, but these go sometimes in all comers, frontline setting, I think that there's no way around it. I mean, I think that maybe they're a smidge low. Maybe they should be 20 to 30%. And maybe some of these other drugs up there, the ones that are in bold are the ones that you're allowed to choose. But, Doxorubicin, that might arguably be a little bit better. It should be up there, particularly if people haven't gotten it. I don't know because they're not telling me how many people got it. But I got to concede, it's a very active drug. There's no doubt about it. This is an active drug. Let me say that again. This is an active drug. I do think it will have a role in this cancer. But, should it be your go-to, you have a hormone receptor positive patient in front of you. I think we'll come to this in the conclusion. You probably need to maximize endocrine therapy. First, you need to give some chemotherapy. And when do you want to reach for this? Second chemotherapy? Third chemotherapy? How much did you really push the endocrine therapy? I think these are important questions. The ILD. The ILD, it's no joke. It's no joke. 12% ILD. The ejection fractions look to me a little bit lower on the experimental drug. Nausea, 73%. And this isn't a clinical trial where these patients are getting exquisite management and care for nausea, and they're coming in with 73% anti-grade nausea. This is no joke. I mean, this has some real toxicity. And I think when you start drifting from the incredibly curated cohorts of clinical trials to the everyday Americans that we'll have to take care of, I think these toxicity issues may play a big role, especially if this wants to claim all this market share. Cost. Michael Lamb, pharmacist. This is a good guy to follow. I think he's had some sharp tweets. Destiny Breast 04. Trastuzumab, Deruxtecan, 100 milligram per vial, $2,909.40. At, average body surface, this mg per kg, 75 kilograms, 405 milligrams, which is 5 vials if you don't round. We're talking about $15,000 per treatment. That's not nothing. And how many patients are there that fit this bill? Budgetary impact, this is going to blow the budget. And 12% ILD, how much will ILD management count? Yeah, and the paper said something like, we were very proactive about early detection of ILD. You do know that early detection of toxic side effects is not always the best way to interdict upon them. It's a testable hypothesis, but I haven't seen validation of that. But he's right, it's going to be incredibly costly, and it can come with some fair bit of toxicity. I saw that this Pedro, I'm the only one who liked it. I'm the only person who saw it. And I only saw it when I was like, when I put together this talk, I try to Google slides so that I can easily import them to my spreadsheet, but I didn't find any good slides. But I found a good question. Some questions for someone who's watching from afar. Is it practice changing for triple negative breast with only 58 patients? Good question. I think that it's weird. It's weird that they just included this. I don't see any reason why there's a differential interaction there, but it is really weird. And we do have Secukinumab govitecan, which I have other issues with in a paper I wrote with Timothy. But I do think that we'll have to maybe put a pin in that, come back to that. Only 18% received taxanes. Where did he see that? I've been looking for that. I couldn't find that exactly. And anthracyclines, and I want to know that as well. This is an issue since their response rate tends to be higher. I think what he's saying is that 18% of people, their investigator choice was a taxane, because I'd love to know what they got before. And the reason they might not be getting taxanes as much now is that they have just progressed on a taxane. That would be a good reason, but I need to know that. How about germline BRCA? Did they receive platinum? And that's an important question. Did they? He signed it best. I like to... it's sweet when you see people sign a tweet. Conclusions. My overall conclusions. And again, I'm in the dark here. How can I judge a trial? I know nothing of what they got before. I played my little boat game with you. I subtracted. I told you a little bit about the triple negative breast cohort. They're getting, as you know, the standard of care therapy of endocrine therapy and cyclin 4/6 kinase inhibitor. I don't know why they're getting it, but it makes you wonder. Makes you wonder about what's going on and what are the resources. Some of the places they're enrolling patients. It does make you wonder. Makes you wonder if your post-protocol therapy is actually up to snuff or if it's just kind of a bon voyage send-off. This is a very active drug. It's very, very active. One thought I had is, this is a question for the audience, please tell me, does anyone know what happens if you just take the payload and develop the payload? I mean, they keep talking about this bystander effect, and of course all the evidence I see for that is a cartoon they've marshaled, but this drug also has activity in, I think, totally HER2 absent disease. Is it possible that it just goes in, the payload dissociates somewhere, and then it just starts, boom, killing cancer the way topoisomerase inhibitors do? If so, what would happen if I just, developed the payload? Has anyone tried to develop the payload by itself, naked payload? I know we love, precision, everything's precision, but is it possible that the naked payload would do pretty good, maybe at a different dose? Does anyone know about the history of this drug, 1997 payload? Obviously, when you tether it to the antibody, you get a patentable compound, but is there any development history of the payload? What happened to people before and after? I don't think you can really assess a study until you know about post-protocol care. And who are the people who enrolled in this study? How do you get into the study in hormone receptor positive? You had to have gotten endocrine therapy in the metastatic setting. You had to have gotten 1 to 2 cycles of chemotherapy, but you only have 1 line of therapy. How does that work? You had to have gotten that in the adjuvant setting, right? The chemo at least, but not the endocrine because that doesn't count. And then the endo, plus or minus 4/6, and then you had to enroll. I mean, is that right? Am I missing something? We need to know that. How can anyone give standing ovation and not know? You want to live you want to live in the dark? Anthracyclines, platinum. Why restrict the choice? I mean, we're getting to 5 investigators. Why not just give a truly unfettered choice? Can we finally do investigator choice and it'd be a real unfettered choice? Pre-treatment information released, like all that endocrine therapy in hormone receptor negative patients, and the CDK4/6 inhibitor therapy in hormone receptor negative, it really makes one question what's going on in these locations. What do they have access to? And that's really relevant for post-protocol because if you don't have access to anything post-protocol, a drug that improves PFS is also just more time on drug. And when you progress on the control arm in the real world, you get a different chemo, but here you might not get anything at all. And so that might even shorten overall survival on the control arm more than the treatment arm, which has a longer PFS, which is more time with chemotherapy. And there's some stuff I've written about this in the book Malignant on the part on Cleopatra. In the real world, I do think the benefit will likely shrink, both because people will have more aggressive disease. Trials, of course, obviously select for more indolent biology, and also that some of these things might play a role. Of course, it was written by a medical writer. God forbid anyone write their own papers these days. The toxicity, I think, is going to be formidable. It's going to be a real deal in the real world. I don't think they've fully solved this problem, but it is active, and I do think it will have some. I do think it ought to have some role. I'm just not totally sure what that role is. Total guess, but we got 17.5 going to 23.9 months, 6.4 month improvement in OS. Total guess, but I wouldn't be surprised if a real world is about 9 to 12 that you're going to get out of this. And nobody was blinded except the reader was blinded. The reader was blinded to what they got before and after. Thanks to Dr. Timothy Olivier for looking at this as well. And I took his table because I was about to make that table, but I didn't. So, what are my closing thoughts here? My closing thoughts. Well, low HER2, huge market share, super expensive drug, ILD, super expensive. We're going to spend a lot of money on this. I'd love to see $1 per quality cost effectiveness analysis. I wonder what they got before. I wonder what they got after. Another point is, can we see a breakdown of survival based on whichever one the investigator chose? Did eribulin do better than paclitaxel? Did eribulin do better than nab-paclitaxel? What about GEM? What about Xeloda? Did Xeloda do the worst? I'd love to know what people got before. Did anyone actually get Xeloda, post-neoadjuvant for positive path residual disease? Did anyone get anthracyclines before? Is there a reason why we're not giving anthracyclines? Did anyone get platinum? These are questions I have. Why does somebody hormone receptor negative get hormonal therapy? These are eternal questions that may someday not be answered. Destiny Breast, I think, I think it just requires a lot more probing. I mean, this is an opening salvo. I see a glimpse of what it is, but I don't have a sense of what they did. Standing ovation, I think, is wrong, but also it's not going to get the shine treatment on this channel. I'm not going to say this is at the outset a crazy thing to do. I don't see anything. I don't see any. I mean, except for the fact that some people got some strange pre-treatment that you can debate. I don't see any real red flags here, and I think it is, possibly persuasive. It's super active. I mean, that response rate has blown it out of the water. So they don't have any reason to hide. They can tell us what went on. Well, I'm sorry. This is the best you're going to get with maybe 2 hours of work. So, Destiny Breast 4, my thoughts on it. Just some thoughts, how I think about the paper. We've covered so far in our ASCO plenary series, we've done Shine, we've done Dynamic, which Dynamic was the one video I see a lot of people say, before I watched your Dynamic video, I was gung-ho about it. I watched Dynamic and yeah, you changed me. Shine, I hear a lot of people say, why are you criticizing us for running a totally useless study? I mean, I don't know what to tell you people in Shine. Come on, you knew it was bad. I also got another interesting point on Shine, which is like, why did it take you so long to report these outcomes? Why did it take you so long to report it? You could have reported it sooner. What was your p-value doing all these years? I want to know. We did determination, as in we're determined to find a reason to keep transplanting, even though transplant is on the ropes. And I talked to a myeloma expert who said that it ain't looking good for transplant. And now we're doing Destiny Breast 4. I really want to do the Luis Diaz paper on dostarlimab, or that PD-1 inhibitor for locally advanced rectal cancer. I really want to do teclistamab. I think that's an interesting bite that's coming in myeloma. And I think that anything else, put in the comments, email us if you have suggestions. I'm going to do that. If you're a fellow doing Journal Club, I hope this was good preparation to you. Good luck. And that's all. Until next time on the Plenary Session Podcast, follow us for the most incisive oncology dissection. Read the book Malignant if you want to be a better reader of oncology trials. And until next time.